Hi, I'm Elena, a third-year PhD student at Newcells. Every year, 4 million animals are used for animal testing in the UK. Whenever a new drug is developed by pharmaceutical companies, it must undergo testing for safety, efficacy, and toxicity on several different animal species, such as rat, mouse, and fish, before it can be used on the human in clinical trials. Since treatments are designed to cure human diseases, this condition needs to be induced in the animals used for preclinical drug testing. In biosciences, inducing a disease in a healthy animal is called modeling. The most common diseases in humans are chronic, so when modeling such condition in an animal, we need not only to injure whichever organ is affected by the disease, but also to make sure that the sick animal stays alive as long as possible, so that we can make a comparison with what would happen if we treated an 8-year-old sick human with that drug. The trade-off for the animal involved in the testing is huge. Once the disease is induced, they're going to be chronically ill, definitely unable to ask for help, and possibly in pain. Kidney diseases are a prime example of smooth killer chronic illnesses. Patients do not realize their kidneys are not working well anymore until it's too late to treat them. And at this day and age, the only two options for a patient with chronic kidney disease are dialysis and transplant. So pharmaceutical companies are making a big effort to develop new treatments for these patients. This means that many animals are being used to find the right drugs to cure the disease. The aim of my PhD thesis is to create a three-dimensional model of one of the kidney segments called proximal tubule by using human cells harvested from kidneys that cannot be used for transplant. This miniature proximal tubule can be injured to model chronic kidney disease and can be used to test how safe and efficacious new disease treatments are. To make sure that the 3D kidney proximal tubule model can actually be used as a substitute for animal testing, I first need to prove that my model carries out all the function that a real proximal tubule is able to carry out on a daily basis. So what does a proximal tubule do? As its name says, it is a tube composed by a very special type of cell. Its job is to get rid of nasty molecules by moving them from the blood to the urine, at the same time as retaining good molecules such as sugar and proteins. Proximal tubule cells are also busy talking to their neighboring cells, whether this is to warn them that they are stressed, to let them know that they're happy, or that they're not feeling so well. The proximal tubule cell's best friend and confidant is a cell called fibroblast. The fibroblast not only listens to all that the proximal tubule cells has to say, but also changes its shapes and behavior according to its best friend's mood. If a proximal tubule cell is upset, a fibroblast will be upset too. This will lead to a cascade of unpleasant events that will turn the surrounding environment from healthy to misfunctioning. This process is called fibrosis. Fibrosis appears to be the process that leads to deadly chronic kidney disease, the disease that does not yet have a cure and requires the sacrifice of so many animals to be resolved. This is why there is the need of a 3D proximal tubule model for drug testing, for a world with happier animals and healthier kidneys.